Well, good afternoon, everyone, and welcome to the Front Row. My name is Jamie Williamson, and I'm the Executive Vice President here at Scripps Research. Today, we are welcoming Cavalieri, Marisa Roberto, as our speaker, and she's going to be telling us about alcohol and addiction and the molecular basis for that and how to treat it. And I do want to say a few things because her program is part of a very special center here at Scripps Research. And we have a variety of faculty here that are involved in multidisciplinary research. And Marisa is one of a great team that's part of the Pearson Center for Alcohol and Addiction. And this Pearson Center was founded by Barbara Mason, who's a faculty member in our molecular medicine department, and George Koob. George was at Scripps for many years and now is on leave from Scripps. He's actually the director of the National Institute on Alcohol Abuse and Alcoholism in Bethesda. So it's really great to have a strong proponent for alcohol research at the NIH in the NIAAA. So the Pearson Center has a number of faculty in addition to Marisa and Barbara. There's a group of faculty studying addiction, and we have Candace Conte and Remy Martin-Farden and also Eric Zarilla. And there are two adjunct faculty members that are over at UCSD that are part of the Pearson Center, and that's Olivier George and Mike Teff. So this is a really terrific team. They have, in addition to the center, they have a center grant that just recently we hope is renewed that focuses on alcohol research. The Pearson Center was founded by Mark Pearson, and he has a very personal connection to this cause. And I had the occasion to listen to him talk about this commitment at one of the annual meetings for the Pearson Center. So it's really terrific. Mark is also on the board of directors. I hope Mark is logged in, I'm pretty sure. And it's really terrific to have someone this committed to a particular cause and a disease that is a philanthropist, that is bringing in additional resources matched by the energies of our faculty to bring in grant money. So thank you, Mark, very much. So let's go on to Marisa. So Marisa is studying addiction and alcohol, and addiction is basically it's a neurological disorder that causes you to interact with substances in an unproductive way. And fundamentally, it's a behavior that you undergo that's counterproductive. And as such, that behavior is seated in the brain, and the brain is composed of neurons, and all the neurons are interconnected by all of these synapses that make the neurons talk to each other. And at those synapses are chemicals that go back and forth between the cells, and that gives you the opportunity to come up with some kind of medicine that can alter the neurotransmission between synapses, the neuron function, and how the brain responds to various cues from the environment, like drinking. So it's really, you're going to see Marisa kind of drill down in on that. Now, one thing that I would like to note is Marisa, she's a terrifically funny person. I'm fortunate, her lab is across the hall from me. Very energetic, funny person. And one of the things I've learned is that despite being Italian, which is one of the great wine regions of the world, Marisa does not drink. And I find this to be a little bit alarming. I mean, I'm not sure how I would feel about a rancher who didn't want to eat steak. So, it's just, you don't really need to drink in order to understand how the brain functions and how that can lead to addiction and neurological disorders. So with that introduction, I'm going to let Marisa tell you all about her fascinating work in this really critical and important area.
Thank you, Jamie, for the very kind and generous introduction indeed, and for emphasizing that I don't drink. I don't like the taste of it. And actually, let me start saying that you shouldn't be worried. A glass of wine is good for you, because what I'm going to talk about today is alcohol addiction and the research I've been doing here at Scripps. And I'm really excited about sharing that with you. So thank you, everybody, for tuning in wherever you are. So I will want to introduce addiction, in particular alcohol use disorder, because alcohol is the drug we study the most. And I will use alcohol and ethanol interchangeably. So just keep that in mind. So as Jamie already said, addiction is a brain disorder. As such, I will introduce the brain region that we have been studying for several years, the amygdala, as a key mediator of the pathological state that I'm going to talk about it. And then I really want to touch upon two main lines of research: the recruitment of stress system, which is a line of research I started when I joined Scripps, so a long time ago, but still is very exciting and really provide a lot of new avenue to study mechanistic this pathology, this brain disorder, and then try to alleviate the people that suffer of it. And then I want to talk about one of the new direction in our lab that we have been studying, the recruiting of the neuroimmune signaling, because I will show you how this system also have an enormous impact on the development of addiction. But what is addiction, and really why number it matters? So for addiction, I mean alcohol and other drug, illicit drug, and it is basically a medical condition that is characterized by impaired ability to drink alcohol or take the drug. And this happens because there is really changing in the brain that occur. And it is the leading cause of preventable death in the world. And if you put that in the context of the United States alone, you can see that is a very common disorder. It affects almost 50 million of individuals every year. And if you look at the alcohol, that is the drug we study the most in the lab, it almost equals another devastating disorder like cancer. So it's also very costly, because of cost to the society about 700 billion every year. And here you can see the breakdown between alcohol, illicit drug, and tobacco. And again, I like to make the comparison with cancer, and you can see the number very comparable. And for cost, what we mean is the direct cost and indirect costs. So the medical expenses driven by the cost to the individual that suffer of addiction, and also car accidents, loss of productivity, and so on. So domestic value. And then when we put that though in the context of the National Institute budget devoted to study this disease, in particular alcohol, you can see that the budget is very little compared to other drugs and other diseases like cancer. This is particularly alarming if you consider that alcohol misuse per se is often comorbid with other mental illness, in particularly depression, anxiety-like behavior, bipolar disorder, and most importantly, when you consume excessive alcohol, it can accelerate the progression of neurodegenerative disorder like Alzheimer's. So most of the time, really, drinking alcohol becomes a coping mechanism to empower the mental illness. So kind of self-medicating to make you feel better. Also, if you have a problem, you know, and you seek treatment, if you have other mental illness, it complicates the potential treatment. And when it comes to treatment, because alcohol use disorder is a medical condition, as I told you, is treatable, but unfortunately, only less than 10% of the sufferer actually seek for treatment. And this is very, very alarming. And why is that? Well, most of the time, talking about alcohol misuse is problematic. People don't feel comfortable to talk about it. So it's often the elephant in the room. It's there, but nobody wants to acknowledge it. And most importantly, another negative fact is that for years, for decades, there has been a negative connotation when it comes to addiction in general. So people that suffer from addiction is resilient to seek treatment because they don't want to go public and recognize the problem. And this, as society, we need to do better. And thankfully, the National Institute of Health and the WHO are really promoting more information to get to the society and let us really understand that addiction is a brain disorder, because it really it's a transition where the brain stops functioning the normal way. It's hijacked, and we need to understand the mechanism and the base of this dysregulation to have a better diagnostic tool and also to treat it better. And this really, I want you to think about this. I show you the number of cancer for a purpose. So the number of alcohol misuser actually, and cancer patient is similar, and the cost to the society are similar. But think about it, only one person out of 5 that suffer IUD seek treatment. Cancer patients, mostly, they all get treated. So that's really a perspective that I want you to pay attention to. And with the pandemic, it has been very clear that the increase of alcohol drinking has been escalated by 35–45%. And we hear in the news always overdose, drunk driving. And there are factors that have driven this escalation in drinking, particularly the social isolation. So the staying home at the beginning and all the shutdown that we had through the 2 years, the loss of job, then the financial insecurity of what really tomorrow brings us, has led to this greater depression and anxiety. So all this uncertainty in life, this event in life, have really promote drinking as coping mechanism with distress that affect us. But after all this negative and really heavy-hearted point, because I bet all of us know somebody that suffers of addiction in a way or another, so it's a problem that really touches our life every day. But the good news is that not everybody is susceptible to develop the problem. So not everybody that just like drink in a social setting develop this problem. And that's really something we need to keep in mind. So continue to drink your glass of wine with your meal, with your friends in moderation. And when you do that, really you feel good, because what alcohol does, like other drug, addicted drug, it acts on some neurochemical in the brain and part of the brain that make you really feel happy and feel good. However, in a subpopulation, only like part of these people that drink socially become more at risk and more susceptible to develop a problem. And that is because they then cannot stop drinking, they cannot control the amount of drink they take and how often they take it. And only 6 8% of these subjects actually develop the dependence. And this state is what we study in the lab in preclinical model. And it is the stage where the brain really stops functioning in a normal way. And there are brain region and specific system like the stress system that I will talk in a minute that are recruited. So in that point, you don't drink because it makes you feel good. It's because when you don't have your drink, you don't feel comfortable. You have a negative emotional state that is characterized by anxiety, irritability, and you really don't feel well. So you drink to feel normal and to alleviate it, to normalize this imbalance that is being created in the brain and in specific brain region. So that brings to the nature of the progressive and relapsing nature of the addiction, that where you drink excessively, you can't control yourself, and then when you have your drink, you are in abstinence, you crave your next drink. So in this chain, in this progression, you can just look back and think of COVID, but also 9/11, all stressful events in life that touch our life dramatically, even trauma of any kind, really add the stress component that is aggravating this situation. As Dr. Koob, George Koob, that I'm glad Jane introduced him, he was my chair when, for most of my career here until 2014, when he went to lead the NIAAA. He is basically the promoter of this framework that is called the Neurobiology of Addiction. And really, thanks to him, I really fall in love. We studied this disease, although I don't drink, as Janie said, because it really touched the life of so many people. So what George conceptualized here nicely is that because addiction, the progression to addiction involve different brain regions, you know, there are different color-coded stages of addiction. We study particularly the negative effect, so the part where you drink to relieve this emotional negative feeling that you have that is anxious and is like malaise. So what we call that is the dark side of addiction. Where stress component, so stress system in the brain, particularly in this structure here called the extended amygdala, that comprise also the CA, the amygdala part that I study in the lab, is really the devil component. And keep in mind, this is the stage we study where there are a lot of factors like genetics, childhood trauma, psychiatric comorbidity, like with the mental illness, that aggravate and keep this relapsing circle going. So at that level, there are reward systems that are down-regulated, but the stress systems that are up-regulated. And with this in mind, we got interested in studying the central nucleus of the amygdala, so the CEA. The amygdala, as I show you, is part of this red part, the devil part of the addiction. And it's one of the most conserved phylogenetic brain region across species because it mediates really primitive instinct like fear, stress, and anxiety. So it's the perfect brain region that can we study in an animal model, particularly with mice and rats. A couple of things I need to emphasize: that brain region contain neurons that produce neurochemical. The major neurochemical is the GABA signaling. GABA is the main inhibitory neurochemical in the brain. But this neuron also produce little protein called neuropeptide, and particularly the peptide that mediate the stress responses that I will discuss in a minute. And a lot of the work by George and others here in the Alcohol Center all over the world has shown that manipulating the stress system and particularly the GABAergic signal could affect and alter behavior in the animal model. So with that in mind, my mission when I came here to do the postdoc was really to look at the basic cellular mechanism and synaptic mechanism, as Jamie mentioned. So what we do, we use the animal model, we treat the animal for a long time, like several weeks, such as they take ethanol every day for weeks, mimicking basically the dependent state. So we make them addicted on alcohol. Then here I'm showing you a nice section of the brain of a mouse, and the amygdala is here. Here I show you actually a brain slice. So when these animals are dependent, we always have, of course, the control animal that they haven't drunk any ethanol. What we do, we anesthetize them, we get the brain, we slice it, and we keep the slice alive for 10 hours, and then we record from this neuron alive. So to put that really in a more simplistic manner and showing you the synapses, which is the point of communication between two neurons in the brain, wherever brain region it is, but in the amygdala, I told you there is a lot of GABA, so you consider there is a neuron that is communicated to the next neuron. We conventionally call this neuron presynaptic side. What this neuron uses is this chemical, the GABA, that is in vesicle, but under its electric stimulus, basically get released in the synaptic cleft. Then on the receiving neuron that we conventionally call postsynaptic neuron or postsynaptic side, you have this little protein, those receptors that can bind the GABA and they transduce the signal. So the flow of information goes in this direction. So how do we measure that? So I told you we have the brain slices. We really poke the brain. So what I mean by that, we have a glass pipette that we put in the slice and we record from single neuron. So what we observe are these little events, you know, and what we quantify measuring is how many of these GABA currents you have in a given time. So one thing you need to keep in mind is that if we give a drug, we can see really right away after a couple of minutes the effect. So if you see an increase in the frequency of this event, what it really means is that the GABA release at presynaptic level is increased. And keep that in mind because I will show you that's what ethanol does when we apply that on the slide. Increase communication at GABA level across neuron. We also measure other stuff, of course, but one of the most beautiful things in being an electrophysiologist is that, first of all, you have a piece of brain that is alive. While it's from animal, but that's, you know, you can see right away the effect of a drug that is highly rewarding, talking about rewarding. So now, those neurons also use a lot of peptide. I told you, particularly the corticotropin releasing factor is the peptide that regulates the stress response. So those neurons in the amygdala produce this mediator of the stress. And also here, just to schematize how it works, is like this is the ligand, this is the peptide. There are different receptors that can bind. So those proteins that would bind the ligand, when it comes to alcohol use disorder, the CRF1 receptor subtype are the one making a lot of the job here. So that's why I'm focused on that. And George Koob and other group, what they had shown is that if you block, so with the drug that antagonize, so block the binding of the protein to the receptor, what you see in the animal, especially in the dependent animal, is that they relax. There is less anxiety, like behavior, they drink less, and those are all the phenotypic expression of the pathology that we study. But we didn't know at that time when I started working on that, what was really the mechanism. We knew in the animal the symptomatology of drinking and anxiety would be decreased, but we didn't know how that happened. So when I started my postdoc, now 20 years ago, I came to Scripps, and I have to acknowledge for a moment George Siggins, my mentor, because George is a brilliant researcher. He has supported my career through the years so much. And one thing I learned from George is that, and I am really thankful, he always gave me the freedom to do the experiment the way I want it. And I am immensely grateful to George, really, as a mentor. So one of the first questions I wanted to answer was whether this GABA signaling in the amygdala that is so involved in the dependent state could be changed when the animal had received ethanol for several weeks, so was in the dependent state. And here sometimes I use chronic intermittent ethanol exposure just to facilitate, but those are dependent animals. So one thing we observe is that in the dependent animal, the amygdala has a lot of GABA. You see, look compared to the naive control animal. The frequency of these spontaneous iPSCs is almost double. And what I told you earlier, that means there is more GABA being released in the pathological condition. Then another question we wanted to answer was whether we apply acutely ethanol on the slice from both naive and dependent animal, could alcohol do something at those synapses? And that is an experiment that seems trivial, but what really mimics in humans is the acute intoxication. So when you drank too much, you get intoxicated. So what we found is that in the amygdala, what happened in both conditions, so in both naive and dependent, what ethanol induces is an increase in GABA release that is very comparable. And that was very striking in the alcohol field, because that meant that there was lack of tolerance. So you didn't need a higher dose of alcohol to induce the same effect. But most importantly, we wanted to understand if the CRF1 receptor play a role in that. And surely enough, when we block the CRF1 receptor with antagonists, with this molecule I show you, they block the binding, the ethanol effect were gone. So obviously I summarize a lot of my postdoctoral work in 3 graphs, but that really took a lot to understand all the mechanism in place. And recently, I was really gifted to collaborate through the Neuroimmune Consortium with Kathy Grant in Oregon, at the Oregon National Primate Research Center. And I had the opportunity to study non-human primate and to understand if this change in the brain, in the amygdala, is something that occurs in monkey as well. And sure enough, it does. You see, in this amygdala from a monkey that had drunk for several years compared to the control animal, there is a significant increase in the frequency of the spontaneous iPSCs, meaning there is more GABA at those synapses. And one thing I want to mention, actually, we always have done this experiment blind. So we never know what kind of animal we get until the end that we break the code. So we were also very excited, actually, to learn from a great science paper from Marco Eilig's last year in Science, where he also identified that the GABA dysregulation occur in amygdala of human, and that was done using post-mortem tissue of the amygdala. So that really points to the translational relevance of this change in this neurochemical in this brain region that is very conserved across species. So the next question was also to understand whether the stress peptide could do similar thing. as I show you that the CRF1 receptor mediates the ethanol effect. So I did some of this work when I was a young assistant professor. Many of my postdocs, like Raleigh, Rodriguez, and Dean Kirsten in the lab, have reproduced and add always more information to the puzzle. Dean actually is now a professor at the University of Tennessee Health Science Center, and he has his own laboratory. But the thing that we found is that CRF, applied on the slide, it does increase GABA release in naive and dependent animal. And if anything really in the dependent, this effect is larger because we had this recruitment of the stress system. Then we were also very pleased to translate this to the non-human primates, where you can see ethanol induce an increase in GABA signaling in both control and drinker. So another piece of data that has a translational value. So this is all happening in the amygdala on mice and rats and monkey. And but as I mentioned at the beginning, you know, like the brain and in the framework of the development of addiction, the brain is all connected. So I like to show this picture because this is like an electric pole. So that's the amygdala that send a lot of information to other part of the brain, but also receive information for other part of the brain. And in human connectivity diagram here, you can see how complicated it is. And obviously, we cannot just poke a lot of brain region, but there is a big advantage in the neurobiology these days. So we can utilize a lot of transgenic mice. And the way this is really enabling us to understand more specific neuronal population. In particular, of course, I am interested in the stress system and how the circuit from the amygdala goes to other brain region. We use these mice and it's easy to identify specific neuron in the way they connect, as you can see here. So although I will not show you all the data that we have accumulated through the years, what I want you to understand is that the GABA compromised GABA system in the amygdala is basically something that is happening in this brain region, but it translates to the whole brain. So what happened in Vegas basically does stay in Vegas. So from the amygdala, it gets communicated to other brain region, to effector brain region, for instance. And this work has been done in collaboration with Candice Content here at Scripps. We do a lot of this circuit manipulation. And Melissa Herman at the time was one of the postdoc, a stellar postdoc, now a tenure track at UNC. She really exploited specific pathway that were actually critical in mediating the anxiety that we observe in the animal. But we also are looking at how cortical brain region send the information to the amygdala that is dysregulated. And this current work in the mice and rat, where we can really trace the specific neuron, as led by Pauravi and Roman in the lab, two other very talented electrophysiologists. So let's bring it back to summarize this part to the synaptic level. So I show you that when we are in a physiological state, so the equilibrium state, the synapses in the amygdala use GABA, and it gets released, it gets binded, and the signal flow, you have the signal here going. But when you challenge the animal, the slice with ethanol, whether it's acute or chronic, to mimic the dependent state, what you have is an increase in the stress system. And then this mechanism is enhanced. You have more GABA release. So there is pouring GABA at these synapses. And then that brings to the IUD, the condition that we study, where there is imbalance. You have way too much GABA and way too much stress peptide. So if we drill down to the mechanism, actually, I've shown you the synaptic mechanism, but of course, a lot of my work through the years has been done to understand how that really happened. So I just want to emphasize that I like to use the red background because we study the dark side, so the devil part of addiction. And we have characterized intracellular pathway that really make the release happening. And of course, we have not only characterized the heteronym CRF, we have also characterized a lot of other neurochemical that are endogenous in our brain. So those are systems that we know and we have demonstrated that the chronic consumption of alcohol that leads to the dependent state alter all these systems. And conventionally, we call this side of the medal pro-stress. And the reason why we do that is because they all act like CRF in increasing the GABA release. But the good news is that the body is so harmonic. You know, when you create an imbalance, you have compensatory mechanism that try to rebalance. And this is what we call the anti-stress mechanism. They are, again, different neurochemicals that are in our brain, particularly the endocannabinoid and other peptides, that actually try to buffer and to counterbalance this damage GABA elevation. And in the way they act is actually decreasing GABA and really offering the opposite side of the coin. So you to have a balance. So it is important that we understand how this all is in place, because we want to find more and better target to use as therapeutic, because alcohol use disorder is really a complicated disorder, is very heterogeneous. And yes, there are medications that are approved, but the complexity of the brain mechanism that get damaged is enormous. So the reason then we want to understand the mechanism, for instance, is how we can restore this mechanism. And I'm really proud to have been a collaborator in this study with Ryan Shandy. Ryan Shandy is an organic chemist at Scripps, and he is a brilliant scientist. He basically synthesized GABA mimetic drug from natural products. So, you know, from plants, that's great. In this case, the ginkgo biloba, for instance. And those are drugs that are GABA mimetic. So in a way or another, affect GABA release of GABA receptor. So they were perfect for us to test in our in vitro setting, and also offer the opportunity to really take advantage of the great environment that we have at Scripps, highly collaborative, so we can work with the chemists, with computational research like Stefano, and really synergize. And my goal, my dream is one day to test some of the GABA drug that Ryan developed to normalize these synapses. In the way we did it with gabapentin years back in collaboration with George Koob. So we tested gabapentin, is FDA approved drug for neuropathic pain. It's an anti-epileptic because act on GABA. And what we found is that gabapentin worked through calcium and GABA B receptor, and it normalized this imbalance that we have seen in the dependent animal. Most importantly, then, after we tested in vitro, George and his group tested in several models of excessive drinking. And what we found is that gabapentin decreased the drinking. And that was very important because those results were instrumental for the clinical trial. Then later, Barbara Mason conducted here Scripps testing gabapentin. And gabapentin is indeed a great option to treat a subpopulation of IUD sufferer. So that's why we do this. We want to understand this neurochemical in the brain, and then to find something to normalize and be beneficial in a preclinical setting as well on the human side of the scale. So to summarize this first and long part of my talk, I show you how alcohol and CRF, the stress peptide, act actually in the amygdala, increasing GABA release. And this is something we observe across species, pointing to the key translational validity of our study. Also, I've shown you that if we normalize the GABA CRF transmission in the amygdala that is compromised, is upregulated in a way, we can actually alleviate several aspects of IOD in preclinical and clinical study. And this is very exciting, particularly because lately, I will not show you the data, but we have found some mechanism where the stress system is very intertwined with the neuroimmune signaling that I'm going to talk in the last 10 minutes. So the neuroimmune signaling is very complex. And one thing we have found in the alcohol field is that is the key integrator of both the stress response we have talked about and the chronic alcohol consumption. So the way they do it, I will show you in a minute, but chronic exposure of alcohol stress, they all work in the brain, in changing, in increasing neuroinflammation. However, I just want to mention something and show you the complexity of the neuroimmune signaling as the integrator of those two disorders. It's very complicated. But one thing today we are not going to talk about it because we focus on the brain, that 50% of the liver problem that we have in the United States are due to excessive chronic alcohol consumption. So that's something to keep in mind. So again, we keep it simple, keeping it limited to the brain. And what we, it has been very inspirational in the alcohol research field is this review. I really like to talk about the Fulton-Cruz review because it simplifies the complex alcohol neuroimmune interaction that is very complex. So what Fulton did in this review is summarize studies that were done from post-mortem tissue of alcohol use disorder subject compared to non-drinker. And what he found is that the lifetime alcohol consumption corresponded to brain neuroimmune activation. In other words, the more the individual had drunk through their lifetime, the more neuroimmune activation there was in the post-mortem brain. And one way this review summarized this finding in a simplistic way, so my apology for the immunology listening. One way to measure the chronic alcohol inflammation is measuring activation of microglia. Microglia is the primary glial cell in the brain. You can see from this IB1 staining that in the alcohol use disorder software, there are more of these cells compared to the control. And just for full disclosure, microglia, it's helpful. It really does a lot of good things in the brain in non-pathological condition. It helps the synapses to be pruned and so on. But when it comes to chronic consumption of alcohol, the microglia gets activated and release a lot of protein, so named cytokine, that can be bad for you, and they increase the inflammation. So our question after this nice review was, okay, so what is happening in the brain of our mice when we have made them dependent? Can we see microglia activation as sign of neuroinflammation? Surely enough, it is. So just visually see that in the dependent mouse brain, particularly here it's the amygdala, you have more of this cell, and also the shape is different. So yes, Basically, chronic alcohol increase inflammation as measured by microglia activation. This work is part of the Indian Neuroimmune Consortium. That is an amazing opportunity, and I'm thankful this NIH-funded consortium for many reasons I will talk about in a minute as I conclude this part. So knowing that our ethanol-dependent state involved microglia activation, we decide to see whether decreasing inflammation, so basically ablating, shrinking this cell in the brain would affect synaptic transmission, for instance. One of the things we measure, of course, is looking at the GABA. You can see that the microglia depletion actually decreased, normalized that GABA dysregulation. And we were really happy to see that. So Florence, Rich, and Sophia, they all were postdoc in my lab at the time. They all have faculty position. Florence is at Bingen, Sophia is in Vienna, and Risha is starting her own laboratory at, in Chicago soon. They were really so thrilled to work on this project. So that project also led to another interesting finding. So I told you chronic inflammation in the brain as marked by microglia activation. These cells produce cytokine. Some are bad for you, but some are also good for you. And one of the cytokines that would always show up was interleukin-10, which is anti-inflammatory. The interesting thing that we found, though, and this work was led by Risha, we found that in the dependent animal where we see more inflammation, you have actually less anti-inflammatory element. So that's something that is tilted the needle versus the inflammation because you remove this protective factor. And when Risha tested what it does at synaptic level, surely enough, his cytokine decreased GABA. And I told you, increasing GABA is bad. So IL-10 is a good cup. What it does, decrease GABA. We were then able to collaborate with some of my very strong immunology here at Scripps, in particular Silk Path, and she helped us to understand systemic IL-10 and as well IL-10 in the brain, and we were able to manipulate this system to restore the compromised GABA transmission and behavior phenotype. So to finish this part and bringing back to the communication across neuron, I told you we are in the dependent state, so you have an imbalance. More GABA, more CRF, and now we have a different suspect, the inclusion of microglia. This new cell that actually can release neurochemical protein that can also affect synaptic transmission and contribute in a way and another to the unbalanced GABA. But that brings to what we can do to understand what's going on. So of course, the layer of complexity have grown exponentially, but at the same time, we have found new druggable target. In this case, this cell that produce a lot of cytokine. Again, I put it as the two faces of the coin, where you have the pro-inflammatory cytokine, like IL-1 beta, IL-6, IL-18, they are all cytokine we are working on right now through the Indian Neuroimmune Consortium, and we study how they contribute to the GABA release. Also, we have the anti-inflammatory side of the addiction that try to blunt and oppose this mechanism, normalize this picture. Thankfully, our information at cellular level is always important and the next step for clinical work. And I want to mention a Indian study where we tested Otesla. Otesla is a Primilast, is FDA approved drug, and was tested in several drinking animal model, and it resulted very, very promising in decreasing drinking. Most importantly, in this case as well, Barbara Mason did the clinical trial, and she really found that apremilast can cure IUD in a subset of IUD sufferer, and that's amazing. And one thing I want to emphasize, we are working on other FDA-approved drugs that are already used for inflammatory disorder, and apremilast, for instance, is used for psoriasis by list other, and it's part of the repurposing program that NIH has. And I think this is terrific because will help us to skip some of the long steps to take a drug from the bench through the bedside. So summarizing the last part, I've shown you that the recruitment of neuroimmune system, particularly in the component of the microglia and IL-10, contribute to the elevation in GABA. If we restore manipulated this target, we restore the cellular and behavior phenotype in animal model as well in the individual that suffer with IUD. Then the takeaway message is that understanding the neurobiology of IUD in our preclinical model, whether it's in a slice preparation and then next to the animal model, is a necessary step for screening for potential therapeutic for this devastating disease. It may be a long process to take a drug from the bench to the bedside, but we have done it before with gabapentin. We have been doing it with apremilast, and definitely we have a lot of other molecules that we will test in our study with Barbara. And with this, before I go to thank everybody for listening, I want to acknowledge two things that I haven't talked about. So treatment, we don't do treatment, but please, if somebody wants to look for treatment and understand more about it, visit the NIAAA Treat and Navigator, because it has offered really a lot of opportunity all over the United States. Also, on purpose, I didn't talk about how much is too much alcohol, but you can find those details on the website for NIAAA. And this brings me to the acknowledge. Thank you so much for your for listening. I want to thank my current team on the left and right. They are the current member of my lab. They all are hardworking, brilliant, and motivated people. And we come in the lab with always enthusiasm to study addiction. And I want to thank past member of my lab that I mentioned throughout the presentation, my colleague and collaborator at Scripps, as well as outside Scripps, the core facility, and the least but not the last, and not the least, though, the funding and support from people that really care about what we do, in particularly Shimo family and our chair is generous contribution. So thank you to Paul and Chloe for supporting my research. Then NIAAA for the grant that they have funded through the years, in particularly the INIA Consortium, the Alcohol Center that Jamie mentioned at the beginning, and also the T32 program that I direct here, that allows us to recruit the most talented and motivated postdoc as future in the addiction field. And also thank you very much to the Pearson Center led by Barbara Neeson, and this amazing opportunity we have to promote addiction research and the Department of Defense. Thank you so much. And now time for questions.
Thanks, Marisa. That was terrific. Lots of questions pouring in. I think, one of the ones that has come up the most, it's a kind of a theme, is about the genetic component of addiction. Everyone knows that people respond differently, and you outlined at the beginning that only a fraction of people tend to become addicted, even if they over-consume. So maybe you could just tell us a little bit about the genetics of alcoholism.
That's indeed an excellent point and question. And the genetic component, it does play at least 40%, has a big impact in the risk and susceptibility to develop the problem. And that actually is something, for instance, even that I want to say, even in the selection of the target that we decide to study in the preclinical level, we always base our study on genetic study done on humans suffering of alcohol use disorder, because through the genetic, we would identify, for instance, those genes that are different and they are polymorphic or they are dysregulated in the people that are at risk. And then we study that even with different line of animal that express specifically those genes, and see how they actually genetically, then you see the behavior, they drink more. And we have rats, for instance, that have a dysregulation of the CRF gene. And what these animals are almost crazy. They drink like fish and they are very anxious. So yes, 40% of the gene really impact your susceptibility and make you at risk.
But it's not like there's one gene for alcoholism, like there are some other diseases.
Oh, there are so many.
It's a polygenic trait that's a combination.
It's very heterogeneous, and there are a lot of genes. So, and that's why if somebody has IUD problem through the family tree, it's you can actually do a genetic screening and see the gene that you have that carry that risk for you. But so, it involves more than one gene. That's correct.
I just wanted to ask, this is kind of a question of my own, but I really noticed that you're doing interdisciplinary work, and you highlighted working with Ryan Shenvey, the chemist, and Silke Paust, the immunologist, and a variety of others. And so how do you meet these people? And so Scripps is a wonderful environment, and there's a lot of collaborations going on in interdisciplinary work. How do you actually go meet Stefano Forlì and talk about computation? And
well, actually, this is one thing that I love being at Scripps, and that's why I came as a PhD student. I never left. It's the perfect place where like I think people are so friendly, and just even at the seminar you start talking what you do, what do you know. That's how it really happened with Ryan. We start talking about GABA after a seminar, and that's it is. And I actually hope that Stefano is going to help us to understand how the CRF binding to the alcohol happen. Actually, we have something cooking in that regard too. The Scripps, as you know, Jenny, is an amazing and really fostering environment for collaboration. And there are so many opportunities to meet your colleague on campus.
So another kind of theme that came up in the questions is let's say you're addicted to alcohol, and then you either receive treatment or you cease consumption. So does that reset you to the initial state before you ever had alcohol, or are you somehow still predisposed to the addictive effects based on consumption?
That's another excellent question. Well, the good news is that if you stop drinking, whether you do that because you have the pharmacological therapy or even psychological therapy, there is a combination also, you do normalize some system. Yes, there are some of the changes that occur in the brain that they come back. So they go away when you stop drinking. However, the subject that stop drinking, unfortunately, also are a little bit more sensitive to relapse. And that's really one of the big problems that we have in addiction is the relapse, that sometimes it can just be triggered just to go out with the same friend that you used to go out 10 years before when you were consuming excessive alcohol. The relapse component and this risk component also remain. But the good news is that you've quit, you normalize a lot of the imbalance that the chronic alcohol had created, the damage that had created.
One of the things that you didn't touch on was sex differences in alcohol abuse. So I'm maybe it's different for rats, but what about people? What do we know about that? And
yeah, of course, I voluntarily didn't touch that topic because it's so very complicated. And of course, we all are aware that drinking in women is very different than in male, in the sense the habit per se, but also the metabolism. For instance, a woman with a glass of wine normally really get almost very intoxicated because just the body composition, the way our body is leaner, like there is a different metabolism. So we are more sensitive in a way at one glass of wine, like you guys can drink more, like 3 beer and you are still okay. But for a woman, due to weight and metabolism. But when it comes to actually the animal model, it's something we have been studying a lot. And when it comes to the stress, for instance, and even the drinking, what it does, the alcohol at cellular level is very different. And that is actually it's a current issue because we need to understand more how alcohol and other drugs work in female, even preclinically, because a lot of the work, Jamie, has been always done in male. Then that doesn't translate even in the therapeutic because all the preclinical work was done, for instance, in male. So not accounted for a lot of the hormonal stuff that unfortunately we women, we have. Yep. But that's a really, we study that and it's very different actually. Yes, it's another good question.
Maybe just one last question. We're over time, but there's a ton of questions, but a particularly interesting one was what about the connection of the microbiome? So there's got to be effects just from maybe even helping digest the alcohol. I've heard you can have a condition where you've got yeast that ferment in your gut and make you drunk. I mean, so there's got to be a really complicated interplay between the microbiome and everything that you just talked about.
That's another excellent point, and is on our agenda indeed. But what we know is that the microbiome really plays an important role when it comes to the inflammatory processes that I show you. That's why I really wanted to show the systemic, because we cannot isolate the brain from all the periphery and the gut-brain connection. So we know some of the neuroimmune factor, the immune factor, can arrive to the brain and have an impact. And this is something actually I've been talking with our young here because we want to tackle. And it's really easy to do in a way that because we take only the brain from this animal, we can give the other organ to our, and then to really study how in the progression to become dependent, those changes in the gut influence the brain. So that's something we hope to do very soon, actually.
A number of people asked this question in a couple of different ways, but and you even showed like some of the other addictive compounds like opioids on a couple of your slides, but is it really the same circuitry and is it the same brain region? I mean, is it all in the amygdala, and are these pathways intersecting and related?
That's another excellent question. So the short answer is, of course, when it comes to other drugs, I also have studied the amygdala because I am interested in see if, for instance, the GABAergic signal is changed. And I can tell you, for cocaine, it does. So it shows a similar changing in the GABA that we see with alcohol. Opioids, similar, but more postsynaptic level. But for drug like the psychostimulant that I just mentioned, like the cocaine, no, the circuitry is slightly different. So there are more the rewarding components that are affected the most. So you have to go in different brain regions to see more the effect. But again, for the dark side, a lot of the research hasn't really been done. And what we have done is actually shows dysregulation in the GABA2. But for opioid, I'll definitely look at VTA, for instance, the reward system mainly, because those components, different brain regions, get recruited at different time in the progression. And, it takes less repetition to become dependent on opioid, for instance. It doesn't take so long for alcohol. Like alcohol, you really had to drink a lot for a long time. For opioid, it's enough few times, unfortunately, because the reward system is compromised quicker.
Okay, I think we should stop here. There's a lot of really interesting and some very detailed and technical questions. We have a transcript of the Q&A, the chat, and Marisa will have an opportunity to respond to some of you offline that we didn't get to. And many of you also asked about accessing the slides or recording, and that information will be sent to you. You're registered for this, so you'll get an email with details about that. This has been a terrific educational experience. I learned a lot. It was really inspiring to hear your interdisciplinary research. It's a really important problem, and at the underlying it is really fundamental neuroscience that we're trying to understand. But it's the impact of it in disease and societal effects is really extraordinary. So thanks a lot, Marisa.
Thank you for the opportunity.
Yeah. So thank you all for tuning in to the Front Row. Next Front Row, I believe, is September 21st, and that's going to be Lasse Ajayi, and she's going to be talking about some digital medicine action that's going on here at Scripps Research. So thank you very much for tuning in, and we'll see you next time at the Front Row.